for here, so we'll just get started then. Okay, so per usual, this is probably gonna cover most of what you guys need to know. If I need to update you guys on anything, I'll do my best. I'll do my best to do that. I have a very busy day tomorrow, so I can send out a late announcement. That's fine with you guys. I'll just make sure you guys know everything. You guys send out anything that you guys need to know. But yeah, so let's go over our final exam review. All right, so first topic is gonna be. So we're gonna first talk about the pharmacology and therapeutics of antipsychotics. So even though this topic is referring to antipsychotics, we're gonna first talk about lithium, the John Cade experiments, because you guys need to know what lithium is and kind of like the main theories as to why it works when we treat patients with bipolar disorder. All right, so John Cade, so he grew up around a lot of people with psychosis, mania. At the time, he didn't know it was bipolar disorder. He kind of just knew something was wrong with them. And so when he got back from the war, he was like, okay, well, I want to study these guys. And so what he did is he took the urine from patients with psychosis, with mania, and he injected them into guinea pigs. And so what happened is the guinea pigs got really sick because the urine from these patients with psychosis and mania, they have increased levels of uric acid. So what we know about uric acid is it's associated with increased levels of inflammation, increased levels of uric acid is not good for you. And so these guinea pigs got really sick when they were injected with this blood from diseased patients. And so what he did in a separate experiment is he put lithium in the urine from these patients, so the lithium could dissolve the uric acid, and then he injected the urine into the guinea pigs. And what happened is the guinea pigs no longer got sick. And what people originally thought is that lithium was breaking down the uric acid, but it turns out that was not necessarily the case. But because the guinea pigs were no longer sick, John Cade was like, okay, well, let's test lithium as a therapeutic in humans, and it was effective. Even though we don't know the exact mechanism, we know that it was effective in treating psychosis and mania in humans. At the time, he didn't know it was bipolar disorder, but now with more research, we know it was bipolar disorder. So now we have lithium established as a treatment for bipolar patients. And so we know now that increased uric acid levels are associated is a predisposition to bipolar disorder. Uric acid, high levels are going to be associated with high levels of inflammation. And as we know, inflammation is a big predisposition to bipolar disorder. One of the most common themes we're going to be seeing here is that everything is kind of interconnected. And so even though we used to believe that lithium broke down uric acid levels, we now think it works through another mechanism. And we think it's working, but it's increasing neuroprotective peptides, or it's decreasing inflammation in the brain, which is why we see a rescue of cortical thinning in these bipolar patients. As we know, cortical thinning is a characteristic of bipolar disorder. And so lithium is thought to kind of rescue this or attenuate this. So lithium will increase gray matter thickness in the brain through possibly neuroprotective peptide release or decrease in inflammation. So now we're going on to antipsychotics and anticonvulsants. So we believe lithium kind of tackles the inflammation, kind of increases neuroprotectiveness in the brain. But we also have medicines that kind of target specific neurotransmitters. So in bipolar disorder, we have dysregulation of glutamate. So with everything in your body, there's kind of like this sweet spot. So you don't want to have too much of anything, or you don't want to have too little of anything. That goes with glutamate transition. If you have too much, you're going to have hyperintensities, you're going to have seizures, it's going to cause cell death. And so that's kind of what we see in mania. What we see in depression is we kind of see the opposite. We kind of see in parecognition because glutamate transition is lowered. So the main idea here is depression and mania, we have dysregulation of glutamate transition, and there's not that sweet spot, that we don't have that sweet spot. And so we need medicines that kind of modulate this. And so the same idea kind of goes with dopamine. And so we have dopamine regulation as well. So we need to use antipsychotics to block the increase of dopamine signaling that we see in mania patients. And so I just have it highlighted here in bullet points so you guys can understand. glutamate and dopamine are two big neurotransmitters that are dysregulated in patients with bipolar disorder. And so that's why we use anticonvulsants and antipsychotics to kind of modulate this dysregulation and bring it back to that sweet spot, back to baseline. And so anticonvulsants are going to decrease firing in glutamate neurons. So we're going to have a decrease of excitation, or it can increase GABA neuron activity. Our GABA neurons are inhibitory. So anticonvulsants are going to modulate glutamate or GABA specifically. And then antipsychotics are antagonists of D2 receptors, which is a specific dopamine receptor. You don't need to know what D2 receptors specifically do. You just need to know it's a type of dopamine receptor, and antipsychotics are modulators of the dopamine system, and they're going to inhibit these receptors, which is going to improve symptoms of mania, because in mania, we see an increase of dopamine signaling, increase of dopamine in the synapse. You are going to need to know like a general idea of how to treat the phases of bipolar disorder. So we have three main phases in bipolar disorder. We have the manic phase, we have the depressive phase, and then we have the rapid cycling phase, where we're going like back and forth between the manic and depressive phase pretty frequently. And we like a very unstable mood in the rapid cycling phase. And so for all three of the phases, the first line of treatment is typically going to be lithium. And so with lithium, there's not a lot of side effects like we see with anticonvulsants and antipsychotics. That's why it's a good first line of treatment. It's kind of good across the board for all phases of bipolar disorder, but we all know it's more complicated with that. Sometimes we need the addition of other treatments, like that second line of treatment shown in this graphic here. For the manic, depressive, and rapid cycling phase, we have different second lines of treatments. For the manic phase, right, we have an increase of dopamine availability in the synapse. We have an increase of dopamine signaling. That's why people with mania are so wired. They feel very like they're on top of the world. And so we need to regulate this dopamine. So the second line of treatment for someone in the manic phase is going to be like a atypical antipsychotic. On the other hand, for depression, we're going to see decreased serotonin levels associated with depression. So we are going to want to modulate the serotonergic system. And so the best way to do that are going to be your SSRIs. So this is going to increase serotonin availability in the synapse. So your second line of treatment for someone in the depressive phase is going to want to be something that targets the serotonin system, and those are your SSRIs. Now, for someone that is going through rapid cycling, it's kind of hard to prescribe them an antipsychotic or an antidepressive medication because their mood is very unstabilized. So someone that has mania, they're like, it's stable mania, if that makes sense. Like they're in a manic mood for a stable amount of time. For someone in the depressive phase, they're in the depressive phase for a stable amount of time. Someone rapid cycling, it's back and forth, back and forth. So it's kind of hard to use antipsychotics or antidepressants to treat someone in the rapid cycling phase. So instead, to kind of mediate this high instability of mood, you would prescribe an anticonvulsant as a second line of treatment for someone in the rapid cycling phase. Anticonvulsant can also be called an anti-epileptic. They mean the exact same thing. There's just two different ways to call the same medication. And so what these meds are going to do is they're going to regulate that glutamatergic signaling by decreasing neural excitability or increase GABA, which is an inhibitory neuron, and then it's going to counteract that glutamatergic signaling. Yes.
Why do you use antipsychotic for mania and depression?
So
I thought you want more dopamine to counteract the depression.
yeah, so yeah, that's right. This is that point where I like say like neuroscience is not necessarily cut and dry. Like all depression doesn't look the same. And even though you're in a depressive state, you can still have like those manic episodes or like hypomanic episodes. So in case of that, you will have like an antipsychotic, but it's case by case. And so you'll have like a primary one that you'll want to administer, but then with every person, it depends. Some things don't work. You want to try other things, and yeah, it can get really complicated in the brain with neurotransmitters. But for the main second lines of treatment, these are what you guys should know. And yeah, so it's important you want to stabilize the mood of people that are in rapid cycling. It's very critical to treat rapid cycling phases because these people are more prone to commit suicide. their suicidal ideology is very high because their mood is very unstable. And so it's very critical to treat people when they are in the rapid cycling phase. And so anticonvulsants will help that, they're mood stabilizers. Okay, so one point that I want to drive home for you guys to understand is that just like everything is interconnected, like the way you eat. Your lifestyle, what you eat, the activities you partake in, like all of that is interconnected with some of your genetic risk factors or genes that you have in your body. It's all connected. Now, you can have genetic predispositions to certain mood disorders. So, for example, like if you are genetically inclined to have increased transcription of genes that are associated with pro-inflammation, this will predispose you to mood disorders like bipolar disorder, because inflammation is associated with bipolar disorder as well as other mood disorders. And or if you have genes that are upregulated in your body that have to do with increased neural excitation or increased glutamatergic signaling, this will also predispose you to bipolar disorder, because patients with bipolar disorder have increased hyperintensity. And so certain gene factors can predispose you, but it doesn't mean you're going to get a mood disorder just because you have these genetic risk factors. It doesn't mean, oh, I have this genetic risk factor, I'm automatically getting depression. It doesn't necessarily work like that. The body's insanely complicated and your lifestyle matters a lot. So lifestyle choices or environmental factors can worsen these predispositions. So like if you have an unhealthy diet, so full of like sugary drinks, red meats, full of purines, you're going to increase your levels of uric acid. So if you're increasing your levels of uric acid, what else are you increasing? Your levels of inflammation. If you're increasing your levels of inflammation, this is going to augment the hyperintensities that someone with bipolar is feeling. It's going to augment cell death in the brain. So once again, these things are all connected. If you have an unhealthy diet, if you're not getting the precursors to serotonin, to dopamine, to norepinephrine, you're not going to be making these neurotransmitters that are strong regulators of mood. So your mood's going to be more dysregulated. So these things matter. And like if you have a lack of sleep, your hormones are dysregulated. Say if you're intaking a lot of illicit drug substances and you're dysregulating your neurotransmitter balance, everything's going to become discombobulated, and it's going to augment those genetic risk factors that you already have. So the combination of genetic predispositions and lifestyle choices or environmental factors are really important. These things all interconnect. And then like in addition, like bipolar disorder is also comorbid with several diseases. Like if you have bipolar disorder, you're more likely to have metabolic disease, cardiovascular disease, anxiety, depression, hypertension. Like these things are all very interconnected. So just understand like how inflammation also plays a big role in this, what can cause inflammation, what inflammation augments. So just understand these connections. Okay. So do we have any questions regarding this topic before we move on? All right. So let's talk about genetic and environmental contributions in bipolar disorders. From my understanding, do you guys have a lot of biology background or understand DNA transcription? Like, do you know what I mean when I say DNA transcription? Okay, so I guess, so DNA, right? So DNA is in your nucleus. All of your DNA is going to be found in your nucleus. It will not leave the nucleus. Well, how do you get proteins in your body then? So what happens is DNA is going to be transcribed. So we say we have a strand of DNA, right? It's going to go through this process called transcription. And it's going to be transcribed into mRNA. So mRNA is a thing that actually leaves the nucleus. So mRNA leaves the nucleus, right? Okay. Leaves nucleus, enters the cytosol, and then it gets translated into proteins. And so basically, if you're having less transcription of a DNA sequence or you're having downregulation of a DNA sequence, the downstream result is going to be less protein expression. So I guess I could just write this here. So less transcription We're down, okay, our down regulation. So these two phrases, downregulation of a gene or less transcription is gonna equal less protein expression. Or less protein being made. And you guys don't need to know the ins and out of like how transcription happens, how translation happens, how you honestly don't need to know all this. You just need to know that like genes get downregulated, there will be less transcription of them, and this will cause less protein expression and less protein being made, which is important because in people that have bipolar disorder or mood disorders and they have these differences in their gene regulation, it's going to predispose them to certain stuff, and it's going to increase or decrease certain protein expressions. And so I'll get into the specifics of that into the next slide, but yeah, this is like a general understanding of like the terminology and what I mean by downregulation of a gene or less transcription and what that kind of means. And so, okay. So what controls transcription of a gene? So these are things called like epigenetic factors. So epigenetics are just alterations to the DNA sequence that are going to increase or decrease its availability to be transcribed. So the only epigenetic alteration that you guys need to worry about is methylation. And so methylation is an epigenetic modification that involves the addition of a methyl group. A methyl group is just a chemical. You guys don't need to worry about what it's made of. You just need to know it's the addition of this group. It's going to add it to the DNA itself or add to the histone that the DNA is wrapped around. All DNA in your body is wrapped around little histones in the nucleus. And then there's some DNA that's not wrapped around the histones, and that's the methyl group can add itself to the histone or add itself to the DNA sequence. But the idea with methylation is that no matter where it's added on the DNA sequence or if it's added on the histone, methylation is going to turn your gene off. It's going to lower levels of transcription if it's added to your DNA sequence. So you kind of want to think of DNA as a book. And so methylation closes that book and you can't read the DNA. If you can't read the DNA, if you can't transcribe the DNA, you're going to get less protein being made of whatever protein that DNA encodes for. You're going to get less of it. And then if you unmethylate a DNA sequence, you're going to open the book. You can open the book and you can read all its content. So unmethylation is going to turn a gene on, and that protein that DNA sequence encodes for is going to be made more. It's going to be expressed more in the body. Does that make sense? Methylation, because I know that gets a little confusing for people. Yes.
So basically, when methylation is happening, that means the gene is turned on and there's more protein.
Turn it off. I'll turn it off. Methylation, yeah, it closes the book. Think of it like that. Okay, so you can't. So it's going to turn a gene off. Less transcription of it, so less protein being. Gotcha. Yeah. Think of it. Methylation leads to less protein being made. Unmethylation, more of that protein. And so just some more terminology regarding methylation, because what we've talked about in class is hypomethylation. So this is going to be decreased methylation of a DNA sequence. So if you have hypomethylation of a DNA sequence, that gene's going to be turned on more, and then you're going to have more transcription of that gene, more protein being expressed or made. Hypermethylation is the opposite. So that, yes.
Question in like our exam, do we have to like memorize genes and their like what they do, or will we just be given a scenario if the link was hypermethylated, what would happen?
It's going to be a mixture of both. So there's only very few genes that you guys need to know, and I'm going to go over them in the next slide. But there's also going to be kind of scenarios about like epigenetics or like methylation. So that's why I want you guys to understand what methylation is and what epigenetics are. And so hypermethylation is the opposite. You're going to have increased methylation of a DNA sequence. With increased methylation, the DNA sequence is turned off. You're going to get less transcription. You're going to get less protein being made. And so, yeah, I just reiterated that in this point here, methylation turns off a gene. And then, I don't know, I like the book. I like analogy, it helps me understand it. So yeah, if you guys wanna like learn it as that, you can. Just know methylation turns a gene off. Less protein is being made.
Are we going to be tested on histone modification?
So, well, not necessarily, well, methylation is a histone modification. It's just in case she uses that verbiage, you understand that histone modification and like methylation of a DNA sequence is this like same thing. If you methylate the histone group, it's gonna do the same thing as methylating the DNA sequence by itself.
Got it, thank you.
Okay, so epigenetics in bipolar disorder. So these are the genes that I recommend that you know. It's gonna be 3 genes and they're gonna have 2 main effects. So I'll just go over them. Okay, so the first one, we're gonna have hypomethylation of this inflammatory COX-2 gene. So hypomethylation, right? So that's gonna mean decreased methylation of this gene. So if we have decreased methylation, what are we gonna have? Increased protein or increased protein expression. So if we have increased expression of an inflammatory protein, we're gonna get more inflammation. And so, as we know, increased inflammation is not good. It's associated with bipolar disorder. It's gonna augment those hyperintensities. It's gonna augment those cell death. And so, this hypomethylation that we see in bipolar disorder is most likely a genetic risk factor that's associated with this inflammatory state. And so, in the lecture, you don't have to worry about the Alzheimer's degree, like disease graphs, it was just, the paper was just looking at that, and it's just like, it goes into like another argument that bipolar disorder could be neurodegenerative, it's progressive because we see cell death in bipolar disorder, so that's why they're comparing it to Alzheimer's disease, which is a neurodegenerative disorder that destroys brain matter. So they wanted to do comparisons, but don't worry about Alzheimer's disease, just know the epigenetic factors in bipolar disorder. So we got increased protein expression of inflammatory genes, this being COX-2. And then we're gonna have hypermethylation of genes that are associated with neuroprotection, neural growth. And so this is gonna be BDNF, which stands for brain-derived neurotrophic factor. And the other gene is gonna be DREBRIN. So these are your neuroprotective genes that are associated with neuronal growth, neuronal survival. So if you have hypermethylation of these genes, you're gonna have less transcription of these genes, you're gonna have less of their protein expression, and so you're gonna have less neuroprotection, less neural growth. And so because we're seeing less neuroprotection and growth, this could probably be connected to the cell death that we see in bipolar disorder patients. So yes.
So do we need to memorize each of these like genes?
Yeah, they're gene, well, they're the name of genes, but like, because they get transcribed or like then translated into proteins, like it's the same name. So, yeah.
So is that like something specific that we should be knowing, or should we just know the fact that like hyper versus hypomethylation of these genes are associated with?
Yeah, you should know the basic function of them. You don't need to know too much detail, just know COX-2 is inflammatory, that's bad. BDNF and DREBRIN are neuroprotective, and that's good. And so you're having decrease of the neuroprotective factors, but increase of the inflammatory factors, and which makes sense based on what we know about bipolar disorder, because we see a pro-inflammatory state, we see a lot of cell death, and we see a lot of hyperintensities. So you just want to know that general idea.
So if there's hypermethylation of DREBRIN and BDNF, then we apply the lithium.
Not necessarily. So the genetic factors is not always like, so we look at genetic factors to kind of like understand like just diseases more. It's kind of hard to apply that to like certain medications. So yeah, you wouldn't need to worry about like, okay, if we're having like these upregulations or downregulation of these genes, we need to administer this medicine. Like there wouldn't be a question. Don't really worry about that. Okay, so yeah. So increased protein expression of genes that are inflammatory or proteins that are bad for you, and then decreased expression of proteins that are neuroprotective and are good. We understand methylation. I know it gets a little tricky, so if you guys have any questions, please don't be afraid to ask. Okay, so I just wanted to just have a list of kind of like the main characteristics and symptoms that we see in bipolar disorder, just like the basic symptoms and characteristics. So we have increased uric acid levels in patients with bipolar disorder. So increased uric acid levels. We see increased uric acid levels with a manic state. Increased uric acid levels also associated with increased inflammation. And so men are gonna have higher uric acid levels or tend to have higher levels of uric acid compared to women because men are more susceptible to mania. I mean, it makes sense because men are more likely to have bipolar 1 disorder, and bipolar 1 disorder is characterized by those hypermanic episodes or like full manic episodes. And then yeah, uric acid comes from foods or drinks that are rich with purine. So this is going to include red meats or very sugary drinks that are high in fructose. And then bipolar disorder patients are going to have larger ventricles. Their ventricles fill up with more cerebral spinal fluid, causing a lot of pressure into the brain. So they're having a lot more brain atrophy or like cortical thinning because of these larger ventricles that are putting pressure onto the brain. And then cortical thinning. So people with bipolar disorder are going to have a thinner cortex. And so lithium is thought to kind of rescue this cortical thinning. Yeah, that's a major characteristic of bipolar disorder. And we think the pro-inflammatory state in patients with bipolar disorder and the hyperintensities are directly correlated with this cortical thinning that we see. A pro-inflammatory state is going to augment this cortical thinning as well as these hyperintensities are going to be causing cell death in your cortex. Once again, these things are all interconnected. And then we also see amygdala hyperactivity in patients with bipolar disorder. In many mood disorders, we see a lot of amygdala hyperactivity, like a very common theme here. Like the amygdala is important for emotional and like fear processing. It makes sense that there's a lot of dysregulation in this area in mood disorders. So. And then we have increased synaptic levels of glutamate and dopamine. So dysregulation of these two neurotransmitters is a major characteristic of bipolar disorder. Just make sure you guys understand that and understand the medications that kind of tackle on this dysregulation of glutamate and dopamine levels in the brain.
Could you go back to the last?
Do you have any other questions regarding this topic before we move on? All right. Yes.
Is there like a distinct kind of symptom in bipolarism compared to other mood disorders that will always be present?
That's kind of hard. For the knowledge of this class, that you're not going to need to know that. Honestly, from my own personal knowledge, I'm not even entirely sure. Like mood disorders are they overlap a lot. They look very similar to one another, and that's why we talk about comorbidity a lot because they just look so similar to one another, and that's why we have like there's a good amount of people who are getting administered the wrong medications because doctors will think it's something with this, but it's not because they just look similar. But there's these general characteristics of certain ones. So yeah.
So for bipolar disorder, is there only dysregulation with glutamate and nothing with GABA?
So there probably is. I'm not entirely sure of the research regarding that, but I wouldn't be surprised if there is. Scientists can probably find anything like dysregulation in any neurotransmitter regarding mood disorders, but for the sake of this class, you guys need to know just the specific ones and ones that are like highly researched. And we already okay, we know this is happening for sure. Yeah.
Along those same lines, like are GABA and glutamate directly correlated? So like if you have more of one, does that mean you have less of the other?
Not necessarily,
Oh, okay.
but they do affect one another.
Okay.
Yeah, case by case for sure. Okay. All right, so next topic, anxiety disorders defined, topic 13. So I know she briefly went over this. Please do not worry about the ins and out of every single criteria of diagnosing all of these anxiety disorders. There's so much criteria, like please do not worry about that. Just kind of know the general definitions of of these anxiety disorders and kind of like what differentiates each of them. So the first one we're gonna talk about is generalized anxiety disorder. So this is uncontrollable anxiety. This can persist for days over the course of months. It's very excessive. You're kind of just worried about everything around you. It's not really tied to a specific thing. It's just excessive worrying, just excessive anxiety. And so it prolongs for a very, very long time. And then we have post-traumatic stress disorder, PTSD. So she talks about this a lot in classes. So definitely know what PTSD is. So basically, when you are exposed to a very traumatic event in your life, there's the possibility that you can develop PTSD because it traumatizes you to the point where you have increased anxiety in the future, hypersensitivity to stressors, to triggers because of the exposure to this traumatic event. So PTSD is directly correlated to exposure of a traumatic event. That's what makes PTSD. You can't have PTSD without the onset of a traumatic event triggering it. And then we have social anxiety disorder. So this is a specific anxiety that's associated with like social settings or like, and with people. So people with social anxiety disorder, like scary faces will scare them a lot, but also like neutral faces will scare them a lot. They're just scared of people. So that's like their specific anxiety, the social settings and people. And then we have panic disorder. So this is when someone is getting frequent panic attacks to the point where they're afraid of the panic attacks they're going to start receiving. It gives them anxiety. Because if you get a panic attack frequently, you're going to start becoming scared. You feel the onset of a panic attack and it creates this anxiety. So that's what panic disorder is. And then we have agoraphobia. So this is a specific phobia. She just talks about it in class. It's a fear of leaving a safe space. Like there are different kinds of phobias, and these are like tied to certain things. Like if you have a fear of heights, like phobia of heights, or bugs, phobia of bugs. So phobia is like specific, like very specific fears of specific. So in this case, fear of leaving a safe space, agoraphobia. And then we have obsessive compulsive disorder. So there's two main parts of OCD. You have the uncontrollable obsessive thoughts that are paired with your compulsive behaviors that kind of address these compulsive thoughts that you're having. So say like you need to like shower 10 times a day or else you're gonna like something tragic is gonna happen. Like that's like set in your mind. So that's like an obsessive thought. And like the behavior that you conduct is showering 10 times a day. So something traumatic doesn't happen, but that's not really reality. Just because you don't shower 10 times a day doesn't mean something traumatic is gonna happen. So that's kind of what OCD is. Like you have these uncontrollable thoughts and you have these compulsive behaviors to address these thoughts. And it's kind of like a repetition of like certain, like you have like certain regulations or like you have like a certain, like you need the certain, what's the word? Like a ritual, there we go. Like certain ritual you need to have in order to feel safe. So those are the main like definitions of anxiety disorders that you guys need to know. Any questions regarding those or differentiating them?
Is that really all we have to know for topic 13? Just kind of know like all the different types of anxiety disorders and like their basic characteristics.
Yeah, for the most part, yeah. And then I'll look over everything tomorrow before the exam, but yeah, for the most part.
Oh, do we need to know how to treat each? Or-
No, but there's a general way to kind of treat anxiety disorders in general. You will need to know like kind of like the general way, which I will touch up on. Like, I don't know if it's this topic or the next, but I'll touch up on that. Yes.
Wait, so with the panic disorder, it's like the fear of experiencing a panic attack that kind of like causes the anxiety itself.
Yes, because you're having them so frequently, you get scared that you're gonna have one.
Is panic disorder caused by I mean, like, how is it, like, what is it caused by, like, initially?
So it can be caused by like a multitude of things. The main thing that you need to know about is kind of like your body is perceiving like a lack of oxygen. It's like when you can't breathe, and so like whether that's actually true or not, like there's just dysregulations of circuits that like make you believe like, okay, you're not getting any oxygen, but that's not true. Your brain is like kind of tricking you thinking that's happening. You don't need to know like the details of that.
Right, but it's only one of the symptoms, right? You can't.
Oh, symptoms
I'm saying like that's only one of the, like, are you saying that's the So that panic disorder or panic attacks involve like sometimes trouble breathing because of the panic. Is that?
Yes, but what differentiates panic disorder from the rest, like the anxiety from panic disorders has to do with that, like panic attack itself. So the definitions was just like, so you know how to differentiate the different anxiety disorders. But you don't need to know, like, you wouldn't need to know like the super specific symptoms of panic disorder. There are some like brain differences in people with panic disorder that I will go over that you will need to know, but you won't need to know like super specific symptoms. Just like whatever I go over is what you'll need to know, pretty much regarding panic disorder.
Okay, I was just curious about the cause, like, what brings it on in the first place? Yes, I don't know, okay, that's fine.
It's a lot, there's many reasons why you can have the onset of panic disorder. Like the most is just dysregulations of certain circuits that are like associated with like emotion and stuff. Do I know the specifics? Not necessarily, but just dysregulation of certain circuitry.
Do we know stuff about like CCK4 or rhythm assignment and how it correlates to panic disorder?
Yeah, so there is a study regarding CCK4 that you will need to know about, and I'll go over that. Okay, the neurobiology of anxiety. Okay, so there's a couple ways that there's a couple methods that researchers use to study brain activity and brain activity changes with the onset of certain anxiety disorders. So the first one is going to be fMRI scans. This detects changes in your blood oxygenation. Then we have PET scans. So this is going to measure blood flow or glucose metabolism in the brain. And then we have SPECT scan. So this is going to measure blood flow. So even though these three techniques work in different ways, the main goal is measuring brain activity and changes in brain activity when you have the onset of certain anxiety disorders. And so using methods like this, researchers have shown that patients that have PTSD, they have increased amygdala activity and they have decreased frontal cortical activity. So in many human studies, these are like techniques that are mainly used. And so that's why researchers were able to find out that PTSD patients have these changes in brain activity. So dysregulation of circuits involving emotion, involving top-down control is very common in patients with anxiety disorders. And so once again, increased amygdala, not surprised, people with PTSD have increased amygdala activity. Dysregulation of an emotional region in the So the next two slides are going to cover studies regarding these techniques on PTSD patients and changes in brain activity. So these changes in brain activity that I'm going to talk about, you guys don't need to know the exact specifics of like downstream effects of these changes in brain activity. What you guys need to know are the brain changes, like the changes that happen, that occur. You don't need to necessarily know why, you just need to know that these changes occur. And so the first study that I'm gonna talk about is this fMRI study, is what they did. They did a mask-based study in PTSD patients and normal control patients. So here's how it works. So on a screen, they're getting flashed a face, right? A neutral face with no emotion. And then they, like for a very brief second, a scary face pops up and then goes back to the room. And so it's so brief that consciously you don't realize that the scary face was shown, but your right amygdala, it doesn't get past your right amygdala. Your right amygdala is going to pick this up and it's going to increase in activity when that scary face is shown, because your amygdala is what allows you to react to scary faces or changes in facial expression. So even though it's quick enough for you to not consciously register it, your amygdala will still unconsciously register it and increase its activity in response to these scary faces, the right amygdala specifically. And so what researchers found is that PTSD patients did not have this increased amygdala activity that they had seen in the normal controls. Now, this is because people with PTSD have increased amygdala activity. So when they're shown this scary face for a brief second, there's not a significant change in baseline like we see in the normal controls. The people with the normal controls, they have normal activity of of the amygdala. So when they show the scary face is shown, we're gonna see an increase in activity. But PTSD patients, their baseline is a lot higher. But we don't see that significant difference that we see in normal controls. So basically, PTSD patients have an increased amygdala activity in normal patients. They, upon seeing the scary face, the right amygdala activates. Does that make sense? So the next study is also going to look at the amygdala, but this is going to be looking at the left amygdala. So the left amygdala is important for understanding verbal scripts or like voices and like words. And so if you from class, I don't know if you guys remember, but there's a study where researchers were looking at brain activation in response to a traumatic script, like verbal traumatic script, compared to a neutral script in normal control patients and combat control patients and PTSD patients. So combat control patients are people that were registered to the Army, they were put into combat, like got all the Army training, just like PTSD patients, but they didn't get PTSD. And so they're important controls to study too. So knowing brain activation changes in the combat controls is just as important as the PTSD patients relative to the normal controls. And so this specific study looks at changes in the left amygdala, and looks at changes in the ventral medial prefrontal cortex and the ACC in response to traumatic event relative to the traumatic script relative to the neutral script. And so in normal controls, when they're presented with the traumatic verbal script, they see increased activation of their left amygdala. It's very normal. So this is what we expect to see. Your left amygdala is going to process the traumatic script and tell you, well, something's going on, something bad. And so, but in the combat controls, we see something different. We see a deactivation of the left amygdala when they're presented with the traumatic script. And then in PTSD patients, they're not seeing any change in activation of the left amygdala when presented with the traumatic script, bless you. And it's possibly due to PTSD patients have an increased baseline activity of the amygdala. So when they're presented with traumatic scripts, it's not gonna significantly change the amygdala activity because it's already high. And so these changes in the left amygdala that occurs in the normal controls, the combat controls in the PTSD patients. And one thing to know is just like even in combat controls, we do see dysregulation of brain activity. It just differs from the ones that's happening in the PTSD. You don't need to know like specifically why, just know the brain activity changes that occur when presented with a traumatic script.
Just to clarify, in the combat control subjects, it activated more, but not as much as in the control.
Deactivation of the left amygdala.
Okay.
So for both of the studies that we went over, what we should get from is that PTSD patients always have like high levels of activation in their amygdala in both right and left.
Yeah. And then just know the right amygdala was that's what we see increased activity in the right amygdala with presentation of the scary face. And then with the traumatic verbal scripts, we see the left amygdala. And then know the combat controls.
Can you briefly go over what is the difference between the activation in the combat veterans who is experiencely trained versus those has PTSD?
Yeah, so the combat controls, they had deactivation of the left amygdala when they were presented with the traumatic script. And then in patients with PTSD, there was no change in activation in this region.
But I thought in the review guide, it mentioned how the experienced combat veterans, they also show the activation.
Wait, sorry, repeat that.
There's a difference about whether a veteran is having PTSD or versus a veteran who is experienced or highly trained or highly skilled. And in the review note, it says for those experienced veterans, they also show deactivation.
Yeah, they are having deactivation. So yeah, there's deactivation in this region in the combat controls, the experienced veterans, but they just don't have PTSD. Was that your question? Okay,
Yeah, thank you.
of course. And then we see changes in the ventral medial PFC area and the ACC area. So we see deactivation of VMPC activity in combat controls as well as in normal controls. And then in patients with PTSD, we saw deactivation of the ACC, which probably indicates the dysregulation of that top-down control. I don't know if you guys remember from exam 1, the ACC tells the amygdala to calm down. So we're having deactivation of the ACC in PTSD patients, not necessarily surprised. And so, yeah, know these activation and deactivation changes. Don't necessarily worry about why, just understand these changes and just understand with PTSD patients, we're having an increased baseline amygdala activity. That's why we're not seeing significant changes in activity when they're presented with scary faces or presented with traumatic scripts. You have a question? Does someone? Okay. All right. So some more studies. I know it's a lot. So we're touching on the amygdala again, but this is not in PTSD patients anymore. This is in patients with social anxiety disorder. So surprise, surprise, we're seeing increased amygdala responses in patients with social anxiety disorder. And so both the normal controls and both patients with social anxiety disorder, they show increased amygdala activity to scary faces, but it's just stronger in patients with social anxiety disorder. But something that differentiates people with social anxiety and from normal patients is that people with social anxiety are going to also react to neutral faces. So their amygdala is going to increase in activation when they're presented with neutral faces as well compared to controls. And so only the patients with social anxiety will respond to these neutral faces. And so they also did studies regarding like presentation of aversive or noxious odors and presentation of neutral objects. And they didn't see any significant difference between patients with social anxiety and the controls. So this basically just drives the point that social anxiety disorder has to do with like people with like faces. So only people with social anxiety are going to react to these neutral faces, and it's not just like random objects, it's faces, it's people. And so when the aversive odor it didn't change anything. People with social anxiety didn't react to aversive odors. Okay. So I know that's a lot of.
This is a different study than the previous one, right?
Yes.
Is there a specific name that we should know this study and the last study by?
I don't know, like, they're not necessarily, yeah, there's not necessarily like a main name, just know like the previous two studies that I talked about are like with PTSD, and this one has to do with social anxiety disorder. Just know like what specific disorder the studies are researching. Yes.
So don't they just do the same thing, the amygdala? Like, what's the difference?
The different, so what I
Like
want
the people, is that it? Like, is the difference like the people, like the faces?
The different, yeah, the neutral faces, the reaction to the neutral faces. So yeah, so there's this is what I meant by like a lot of mood disorders they can look similar and show similar changes in brain activity, but there's these like distinct parts of each one that can differentiate them. So in this case, the reactions to the neutral faces. All right, so now the panic disorder stuff. So basically, your brain is basically telling you you're not receiving enough oxygen and you're like having increased levels of CO2 in your body. And so your brain freaks out and tells you to freak out and you panic. And so with your brain telling you that there's not enough oxygen and there's too much CO2 going on, you're freaking out and you're not like, you feel like you can't breathe. But it's not necessarily that you're getting less oxygen, like physically, it's just your brain's telling you. And so that's panic disorder. You don't need to know like the super specific circuits of panic disorder. It's kind of like just like the general idea of just like what panic disorder like is and like what your brain, like what your body is telling you, like how you feel. And so even though panic attacks are associated with like this feeling of like increased levels of CO2, we can also induce panic attacks. So we induce panic attacks with CCK4. And so this is the study that you guys do need to know regarding CCK4. And so this study is really interesting because you would think, okay, administration of CCK4, everybody's gonna panic, right? But that wasn't necessarily the case in this study. Not everyone panicked upon the administration of CCK4. So what had happened was that in all participants that received CCK4, we had increased amygdala activity, and this was equal in both the right and the left amygdala. We didn't see any differences in terms of activation between the right and left amygdala. Both were equally increased in activity upon the administration of CCK4 in these patients. And everyone that got administered CCK4, whether they panicked or not, we saw this increased heart rate. And so how is it that we have patients that are panicking, that are not panicking, but they also have this increased heart rate and they have this increased amygdala activity? So it's because everybody interprets their internal bodily cues differently. And so, for example, one person who panicked, right? We have this increased amygdala activity, we have this increased heart rate. Your mind's telling you we need to freak out right now. And so that's your interpretation of this increased heart rate. While another person, their interpretation of increased heart rate might be like, oh, something's up, but this will pass. It's not a big deal, And so these different interpretations of your internal bodily cues, you don't need to know any specific mechanisms of that. Just know like people interpret these things differently. And that's why in this study specifically, not everyone who got administered CCK4 panicked, even though they had the same sympathetic activation and they had the same increased amygdala activity. And so you can kind of think of CCK4 as like the opposite of a beta blocker. So yeah, that's why it's really interesting. So like how we kind of interpret different things and can react to different things. So the panickers are thought to have a stronger reaction to this increased amygdala activation or this increased heart rate. So that's why they panic, but the non-panickers didn't have this strong reaction, so they didn't panic. So yeah, that's just because they interpreted. Do you have any questions regarding CCK4 for this study? All right, and so in the same study, they also did an fMRI of brain activity, looking at brain activity. And so the patients, they knew there's a point in the study where they knew they were going to receive the CCK4 administration. And what the patients know is that CCK4 is going to induce a panic attack, right? So they're anticipating this administration, they're anticipating this anxiety. So upon anticipation of this CCK4 administration, what the scientists saw is they saw increased activation in the ACC. And so because they saw this, it's possible that ACC may play a role in panic disorders or just anxiety-like disorders, because in anticipation of the anxiety, we had increased ACC activation. So before I move on to the next topic, does anyone have any questions regarding that? All right, so we have pharmacology and therapeutics of anxiolytics. Anxiolytics are basically just drugs or medications that's going to reduce your anxiety. It's a fancy word of saying reduction of anxiety. Okay, so this is what I meant, like, when I said that you guys should know, like, kind of like the general way to treat, like, anxiety disorders. So I know we've talked about a whole multitude of medications. There's many ways to modulate specific transmitters in the brain, specific hormones, all these things, right? So, but the thing that you want to target first or treat first in anxiety disorders, this is in general, is you want to treat the depression. So if somebody's having anxiety related symptoms, the first thing you want to find out is, are they having depressive symptoms? And you're going to treat that first before you move on to treating the anxiety symptoms. And the reason for this is because depression and anxiety are very comorbid. And sometimes the medications that come with treating anxiety can have more side effects than your typical SSRI. To treat depression, SSRI will always be administered first. There are less side effects with SSRIs compared to like other medications, other antidepressants, and like anticonvulsants, antipsychotics, all these other medications that we've talked about, benzos that we'll talk about soon. and other serotonin agonists, like they are safer to use. So physicians will always want to treat the depression first. If anybody's has like anxiety related symptoms, and if they don't have depressive symptoms, then that's when you move on to treating the anxiety itself. So SSRIs first always. And so this kind of just reemphasizes that the SSRIs are effective at treating anxiety disorders in general. You will always prescribe them first if you can, but then you start moving on if the SSRIs are not effective, or if patients are just not having depressive symptoms and you need to move on to other medications. So just know that general rule of thumb, SSRIs first always because they're safer. And so, yeah, this is like general rule of thumb for like administration or prescription of drugs. Just kind of know this generally, you don't need to know super specifics. Yes.
Is the reason that benzodiazepines are used because are they like a second line of medication if SSRIs aren't as effective?
Yeah, so it's kind of like, so for rapid cycling, benzos could be a second line of treatment for rapid cycling patients benzos are definitely not the first line of administration because of the high risk of like abuse and habit forming that come with benzos.
I have a question. So for bipolar disorder, first line treatment will be lithium, whereas for anxiety related, the first line will be SSRIs.
Yeah, that's if they have depression. If they don't have depression, you don't want to treat with an SSRI. You'll move on to like other stuff.
I thought SSRIs are also used for anxiety, not correct?
Yes, in generally for like, but with like they have like with the depressive symptoms, yes.
Oh, so if the person doesn't have depression, it won't help their anxiety. So they wouldn't take an SSRI for just anxiety?
They can, but typically, it's really case by case, yeah, but yeah, for generally SSRIs can help, but it's usually to help with that depressive aspect that's like really comorbid with anxiety.
So it helps with depression more than it does for anxiety.
Yes, but it can also help depression, but typically you'll look at other medications if they're not having like depressive symptoms. Yes.
So what's like the main takeaway of the slide? It's like a lot of words.
Yeah, SSRIs are like the general like treatment for anxiety disorders because of the depressive aspect that come with anxiety disorders. All right, let's talk about everyone's favorite drug, benzos. And so benzos are GABA receptor agonists, which means that they're activators of GABA receptors. It's kind of like a fake GABA like transmission. Like it's not actual like GABA in your body, but it's gonna like imitate GABA transmission. So your body's gonna think that there's this increase of like GABA signaling because benzos activate GABA receptors. And so these are classified as sedative hypnotics. So they will relieve anxiety-like symptoms. So these are your anxiolytics, but you need to be careful with them. If you are taking strong GABA agonists, you could pass this kind of sedation, relaxed stage, and go into anesthesia, paralysis. So you just taking medications like benzos, you just want to be very careful. It's kind of why we stand away from barbiturates, because barbiturates are really strong. And so they could cause the potential for overdose and cause a lot of issues, because now it's not sedation, it's you're not moving and you're not breathing. And so you just need to be very careful with benzos, and they're prescribed very diligent. And so something that scientists had seen in patients with panic disorders, that they had less benzo binding in their brains. And this is because they had less GABA receptor availability in their brains. So researchers think this patients with panic disorder have lower levels of GABA receptor availability due to just genetic reasoning. So for example, scientists believe that in the gene that encodes for GABA receptors, there could be something called a single nucleotide polymorphism, which is just a single mutation of a nucleotide on a sequence in your chromosome. And so because of these genetic changes that we see, the genetic changes in people with panic disorder, they just have less GABA receptor availability. And so these decreased GABA receptor levels are associated with increased prefrontal excitation in these patients. And so something about benzos, they are not as addictive as barbiturates, but they still pose like an abuse habit forming risk. So with drugs like benzos, right, you're having this increased GABA signaling or like in the brain, and it's not normal. And so your brain is gonna be like, this is too much, like we can't be having this much GABA signaling. So what it's gonna do, it's gonna downregulate the number of GABA receptors. And so that's why benzos can be very dangerous if you abuse them, because if you get off of them and your body downregulates GABA receptors and you're off the drug, you're gonna have a decreased GABA signaling than normal. And it's gonna cause withdrawal side effects. And so benzos are they're very dangerous. You have to be very careful when you take them because it will decrease the availability of GABA receptors that you have. So you have to take them diligently. You take them specifically for like events. It's not typical that someone takes them on the daily or like very frequently because of the side effects that they pose and that abuse habit forming risk that they have. Okay, and then serotonin kind of serotonin receptor availability in patients with panic disorder kind of follow the same trend that GABA receptors do. So we're going to have patients with social anxiety. Oh, sorry, no, this is patients with social anxiety disorder, my bad. They have lower levels of serotonin receptor. So they're going to have decreased serotonergic signaling in the brain. So kind of like similar to the people with panic disorder, we have lower GABA receptors. People with social anxiety disorder have lower levels of serotonin. So like we could use things like serotonin agonists can like stimulate serotonin activity. So that's where buspirone comes into play. And so serotonin plays a role in anxiety as well as GABA. And so, yeah, serotonergics will also be prescribed before benzos due to them being less addictive and due to that depression aspect that's very comorbid with anxiety. And then, but we do also have other medications like buspirone that are agonists of serotonin receptors that can also be prescribed if SSRIs are not effective. Yeah, we have this role of GABA. We have benzos that kind of modulate like GABA dysregulation in patients with anxiety, and then we have like serotonin medications like buspirone that can also help modulate serotonin dysregulation that we see in social anxiety.
I have a question. What would be like the most significant difference between GABA and serotonin since they are both in charge of like calming effect?
Sorry, repeat that.
What would be like the distinctive features between like serotonin and GABA since they're both inhibitor or like calming?
Well, I mean, I honestly don't even know the distinctive effects. So, like, you guys won't need to know that. Just know that patients with panic disorder, less GABA receptor availability. Patients in social anxiety disorder, less serotonin receptor availability. And so, for social anxiety disorder as well as other anxiety disorders, so the first choices of medications are going to be those SSRIs, just due to that depressive aspect that's very comorbid with anxiety. And because they're safer, other medications can pose certain like health risks or because of their side effects or due to their abuse potential. So SSRI is going to be your first line of treatment. You want to treat the depression aspect first. And so, but there are other medications that you can use. So you don't need to know every single other like medication that we talk about. I would just recommend knowing like the main ones that I've mentioned throughout the review, like the benzos, buspirone, antipsychotics, anticonvulsants, like know these other medications. And so, and then another famous medication that people use are beta blockers. So this just works by blocking epinephrine. And so They just kind of like calm you down, but like beta blockers, kind of like benzos, you will use for like specific events, you won't use them like on the regular, so you'll take like a beta blocker before you do something that's like very nerve-wracking in your opinion, like for example, like coming up here and presenting, like if I wanted to calm down, maybe I'll take a beta blocker, but I don't know. So yeah, that's kind of like, so yeah, first those SSRIs, you want to treat the depression aspect of anxiety disorders, and then you move on to other medications, kind of like the general rule of thumb. Okay. So do you have any questions about that topic before we move on? Yes.
I don't know if it's like still part of this, like the scope of this topic, but for people who take like Adderall and things like that, would you say those mechanisms work similar to these like anxiolytic drugs or they're different?
They're different in terms of like Adderall is a stimulant, and so it's gonna increase like dopamine signaling. It's gonna make you feel wired. So like when you take something like Adderall, increase dopamine is associated with like mania and like psychosis. So not that you're gonna have mania and psychosis if you take Adderall, but if you take enough, then maybe. So yeah, like something like cocaine will like predispose you more to like mania or psychotic symptoms because just the effects on like increasing dopamine is a lot stronger. So stimulants work on like the dopamine system specifically, and like what can induce symptoms of mania or psychosis. And so that's actually like honestly a good transition to the next topic because this talks about substance induced disorders, and so like abusing substances like cocaine or like other stimulants, like it can mimic like symptoms that we see with mental disorders. And so, all right, so you are like, if you take enough stimulants and you have enough dopamine in your brain going off, like you can be vulnerable to feeling manic symptoms, to feeling symptoms of psychosis. And so mania, psychosis, these are main symptoms of people with bipolar disorder. So they can look very similar, but even though they look similar, if you're like abusing a drug, these symptoms are more like clustered. They're like for a short period of time. They don't kind of persist and they're not gradual, like in mental disorders. Like you will probably have mania or psychosis for like a shorter period of time if you're abusing a stimulant or you're like really intoxicated with a stimulant. And so, yeah, that's one thing, like that's with an increase of dopamine with stimulant intoxication. So basically the main thing, stimulants modulate the dopamine system. And so that's why abuse of these substances can look like mental disorders. And so yeah, increase of dopamine with drug intoxication associated with mania and psychosis. But you could also have withdrawal type symptoms that mimic mental disorder symptoms too. If you abuse a stimulant enough, right, your body's gonna react to that. So the stimulant's gonna induce this abnormal increase of dopamine activation that is not good for the body, and your body's gonna kick in a process to kind of downregulate that, and it does that by decreasing the availability of dopamine receptors. So say you're abusing a drug, right, and then you finally decide to get off it, your body has downregulated these dopamine receptors, and so now the drug is absent, and so now you're having lower levels of dopamine signaling than you normally were because of the downregulations of these dopamine receptors. And so this is going to lead to withdrawal symptoms like depression and anxiety, because now you're having this decreased dopamine signaling when the drug is absent. So that's why people stay addicted, because they feel like shit when they get off of a drug. And so it's just to combat this other process that your body kicks in due to this increase of dopamine upon intoxication of a stimulant drug. So basically, yeah, what goes up must come down. If you have abnormal levels of a neurotransmitter transmission, your body's gonna counteract that because you can't have too much of anything. There's a sweet spot for everything, and if there's too much or too little of anything, your body will always kick in processes to counteract that. So that's the main idea with substance-induced disorders and stimulants, because of how they modulate the dopamine system, they could cause this onset of symptoms that look like mental disorders. So, do you understand stimulants and how the fluctuation of dopamine can cause these symptoms? All right, and so yeah, this is a little tidbit from the little lecture I gave, and so yeah, you are gonna need to know some of these things. I will give you guys a little there will be some extra credit, 2 extra credit questions on the test. So yeah, know these things, it'll help you out. And so So even though stimulants are bad for you, and I just told you guys how bad they are for you, there is being research done into stimulant-like substances for treating disorder, like anxiety-like disorders and depression. And so that's MDMA and psilocybin. So these are illicit substances, but researchers are really fascinated by them due to their neuroplastic effects and their effects on like inflammatory or immune stuff in the body. And so researchers are fascinated by this. They act on the serotonergic system, and they do so in different ways. They have different mechanisms, which you don't need to know necessarily like the specifics about, but they both increase serotonergic signaling in the brain. They mainly work on serotonin in the brain, and they may also work on dopamine, but just not as much as it does on serotonin. And so that's why it's like so appealing for researchers. And then like there's a significant amount of patients with PTSD or other anxiety-like disorders or depression that are treatment resistant to antidepressants. So that's why researchers are studying illicit drugs such as MDMA and psilocybin to treat these guys. Like there's been a lot of success with MDMA and PTSD patients, and a lot of success with psilocybin in cancer patients and attenuating depression and anxiety. So yeah, the mechanisms of these things are still being unraveled and how they necessarily attenuate these symptoms, but I think it's really cool stuff. So it's what I personally study in my lab. Well, psilocybin, not MDMA. And so, yeah, so one thing that researchers believe MDMA and psilocybin are doing are repairing circuits that go dysfunctional when patients have PTSD. We talked about how brain activity changes in patients with PTSD, but we didn't really talk about how like circuitry changes occur. So you don't need to know the specifics of the circuitry changes, but just know like what connectivity changes are being disrupted that involve the amygdala. And so we're going to have disrupted connectivity between the amygdala and the ACC. The ACC is important for that top-down control. The insula and the hippocampus. Researchers believe that dysregulation of these connections like are just like a main part of like the symptoms that we see in PTSD patients. And so just know the amygdala with these regions, the circuitry is just dysregulated in PTSD patients, and MDMA and psilocybin are thought to kind of repair these circuits. So yeah, don't worry about what each circuit does, just know the connectivity changes or circuitry changes that happen. Yes.
So we don't actually need to know like everything about the insula or.
Yeah, just know the circuitry changes, the changes that happen.
Okay.
Yes, the basic, just need to know things.
So the amygdala, and the ACC have disrupted connections, and the amygdala and insula and the amygdala and hippocampus. Okay, thank you.
And so this is just a brief overview of like kind of the different mechanisms MDMA and psilocybin kind of work through and what people think they're doing in the brain and attenuating PTSD-like symptoms, anxiety-like symptoms, or like depression. And so, like I said, they're thought to be repairing these disruptive circuits that I just talked about through the induction of like neuroplasticity and serotonin signaling. Neuroplasticity as you get older decreases as your synaptic connections kind of mature. When you're younger, you have this like critical period in the brain where your brain's really vulnerable to being mended, and like your brain's intaking everything that's going on in your environment, like rewiring itself. And so that kind of stops after a certain time in your life. And so MDMA and psilocybin kind of like opens this window up and kind of rewire. They're thought to rewire these circuits that are disrupted in patients with anxiety and patients with PTSD and depression. And then they also believe MDMA and psilocybin are working, like they have effects on the immune system as well. They believe they're decreasing inflammation by suppressing immune cell activation. So they'll decrease pro-inflammatory cytokines or increase anti-inflammatory cytokines. As we know, inflammation is a huge part of mood disorders. Inflammation is a big factor in developing mood disorders, and it can cause like a downstream effects, like it can cause like augment hyperintensities and augment cell death. And so it's important to tackle inflammation and find treatments that tackle on inflammation. And then they're also thought to upregulate BDNF transcription, so kind of like increase neuroprotective peptides in the brain. And so those are kind of like the main mechanisms researchers think that these drugs are working through, and that's why they pose so much promise as therapeutics for people who are treatment resistant to like regular antidepressants.
I have a question about like the use of psilocybin treatment. Does it have to be paired with psychotherapy or the drug alone, the medication? When patients self-prescribe, does it show the similar effect?
Yes, when you are described, like, you can't get prescribed these drugs because they're not FDA approved. But if you're in a clinical trial with these drugs, you need to go through like a therapy session. Like, you won't be administered the drug and be like, okay, you can go home now and you're fine. Like the trip is very highly regulated by a therapist. You're in a nice environment. Like, there's this therapeutic aspect that comes with these drugs as well, but you guys don't need to know too much about that.
Thank you.
Alright, so this is from what I presented in the last lecture. So I sent the lecture slides out on Canvas. That's why they're not like in the regular modules, in case you guys are confused, but it's just a reminder that lecture 12 was modified by me, and these are the topics that you don't need to worry about for the exam. But that covers mostly everything. Do we have any questions? I know that was a lot of material. I felt like I was talking for a long time. Yeah.
Please send this slide.
Yeah, so I already sent them out,
Sorry, I missed that part.
That's pretty much it, guys. Do we have any questions before we call it? Yeah, another question?
Another question, could you go back to the PTSD? PTSD study on the amygdala.
Regarding the basic or the scripts?
Scripts. Oh, yeah, what is so it says the activity, the traumatic script is prevented compared to the neutral script. What does VMPFC stand for?
Oh, ventral medial prefrontal cortex, yeah.
Okay, okay. And NC, what does that stand for? Okay. Control subjects, okay. Got it, thank you. Do you mind if I just note that?
Briefly, after you're done with this, go over antipsychotics, and what we need to know about that.
Yes. So well, before I show the slide, so basically, antipsychotics, like, they're gonna be good for like when someone's like for treating the second line of treatment for someone in the manic phase, just because of their antagonism on the dopamine receptor. So they're gonna decrease dopamine because we have increased dopamine when someone's like in the manic.
And what would be the first line of treatment?
Lithium, because bipolar disorder, yeah.
Can you go over anticonvulsants?
Yes, anticonvulsants are gonna modulate glutamatergic signaling, so they're gonna decrease glutamate activity or increase GABA activity, and GABA's like the opposite of glutamate, GABA's inhibiting. So it's gonna basically anticonvulsants, aka antiepileptics, are just gonna decrease neural excitation overall in the brain, whether it be through the modulation of glutamate or through the modulation of GABA. Let me show you guys the slide. This is also all on Canvas too.
Or anticonvulsants,
There's someone that does in my treatment,
Second line,
for
okay.
yeah, for bipolar disorder, yeah.
Okay.
I have another question. Why is it that we see deactivation of the ventral medial prefrontal cortex in the control of this combat control? Like, why?
Yeah, in the comment. Honestly, I can't tell you. I have no idea, but you guys don't need to worry about that. Just know that there's deactivation in this region when presented with a traumatic script.
Okay, but like, why deactivation? Or and the deactivation of the ACC and PTSD, like why is it deactivation? I guess I'm just kind of confused.
For that one, okay, so that, I actually, like, can speculate on that one, it's because we're just having in PTSD patients, we're having a dysregulation of the top-down control circuitry. So like basically your prefrontal cortical areas, your ACC, like those are brain regions that kind of like regulate like your emotional regions, like the amygdala, basically telling it what to do or telling it to calm down. And so you're just having dysregulation of that in PTSD patients.
Top-down circuitry, you mean of the order in which the nervous communicate?
Yeah, from the cortical areas to like the subcortical areas. The cortical area being frontal cortex, the subcortical being the amygdala.
Okay.
For an example.
Okay.
We don't have any more questions. You can call it, but you guys are, yes.
Regarding CCK4, so that basically just like induces panic attacks, or what does it do?
Okay. The not so short answer, it induces amygdala activation, it induces an increased heart rate. And it will induce a panic attack, but it also depends on how somebody interprets like the increased heart rate and the increased amygdala activation. So it's like, that's why some people panic and that's why some people. So the people that panicked had a stronger reaction to the amygdala activation and to the increased heart rate, so they panicked.
Oh, okay.
Yeah. I mean, I'm gonna stick around because I need the recording to finish loading, but if you guys don't have any more questions, we could just call it. Oh, yeah, you haven't, yeah. No worry, take your time, I like.
You go back to where was it, this, social anxiety disorder and SSRIs, and there was one. Oh, yeah. It says. Thank you.
Of course.
Agonist means it acts on the receptor. It activates the receptor.
And then antagonist is opposite. Sorry, just to be clear with the CCK